Um, so with the muscular system, you always think of the function and muscle, whether muscle tissue. Remember, muscle system is composed of the system here we refers to as multiple organs. And multiple organs has a similar function. So in the lab, you're studying the bicep brachii and tricep brachii. Each one of those is an organ. Why? Is because each of the muscle, or like the bicep brachii, has multiple tissue, right? So we see that skeletal muscle has the muscle tissue, skeletal tissue. It has connective tissues, and it also has neural tissue as well, uh, and certainly epithelial tissue you'll find everywhere as part of the blood vessel. And muscle tissue. You know, we talk muscle tissue here. We're talking about cardiac muscle, skeletal muscle, and smooth muscle. And I expect you to also learn the histology, right? One of the most distinguished anatomical feature of smooth muscle versus skeletal muscle and cardiac muscle, and that is they have striation. Skeletal muscle and cardiac muscle have striation, whereas smooth muscle do not have striation. And the striation is due to these patterns that we see them as dark light, dark light. Okay, the dark band is also known as the A band, and the light band is the I band. So remember, I talked to you about dark, DAR, and I. So the I band stand for isotropic, mean the same attractions for the dye. So you can see that within the I band, there it's the same color, if you will. Whereas the A band is not the same, so it's anisotropic. Okay, and that is because the edges of the A band is darker than the inner band. Okay, it's known as the H zone. So H comes from the word Hela, Greek mean light or lighter. And as you can see that in the I band, it's composed mainly of this lighter myofilament known as the actin, whereas the A band has both myosin and actin. Okay? But the 8 zone is the lighter, has just myosin. And I started to um, give you some of the definition. What is a sarcomere? Well, a sarcomere is a unit length of muscle, if you will. The word sarco means flesh and mere means length, right? Uh, and another way of defining a sarcomere is a functional unit of the skeletal muscle. Uh, Another definition of the sarcomere is the distance between the two Z lines. You see here? The Z line is in the middle of the I band. So the distance between the two Z line, it is considered sarcomere. And muscle tissue function is to contract. And by contracting, there's a model of this hypothesis known as the sliding filament hypothesis. That means contraction, meaning the actins and the myosins slide past each other. Okay, so um, they're actually sliding past each other, right? And if you look at this, and you can say that during the contraction, the sarcomere length is shorter, but not every region. In fact, the A region doesn't change. The I regions get shorter, and the H zone gets smaller. Right? It's just the two. The H and the I get shorter, but not the A. The A doesn't change, and that's the key. Okay? All right. Now, if you pay attention to some of these questions I asked today, you might see them in exam again. And the question is, what is this network of membrane that you find in the skeletal muscles called? Does anybody know what the answer is? This networks of membrane who the key here is that whose function is to store calcium. What's the answer? Anybody know the answer?
Sarcoplasmic reticulum.
The sarcoplasmic reticulum. Yeah, the sarcoplasmic reticulum store calcium. Okay, that's the key.
That's the key.
What is the membrane of the skeletal muscles called? What is the membrane of the skeletal muscle? By the way, skeletal muscle is the same thing as a fiber.
Oh.
What is the membrane, plasma membrane?
The sarcolemma?
Yeah, it is the sarcolemma. So the idea here is that one of the other ways that you can think of in definition, functionally different between skeletal muscle versus cardiac muscle and smooth muscle, and that is skeletal muscle contraction is voluntary. Okay, that means you tell it when to contract. So skeletal muscle.
But with her,
Come on, please. Mute your mic.
I just did it. Okay, good. So skeletal muscle is voluntary, whereas smooth muscle and cardiac muscle are involuntary. So in other words, you cannot you cannot tell the smooth muscle when to contract or the cardiac muscle when to contract. Let me bring you back to the 301. Okay, in 301, this is a cross section of a spinal cord. And you can say, oh, this is the anterior because the anterior fissure is bigger than the posterior fissure. And this is this area here is known as gray matter. All of this is gray matter. Now, the gray matter tend to have the body of the neuron, whereas the white matter, which are here, all these have myelinated axon. So they provide conduit to and from the brain, for example. So all of this is gray matter. Now, typical, what you see is that the sensory neuron enter the spinal cord at the posterior horn. So this is your posterior horn. But at the anterior horn, typically you have the body of the motor neuron sits here, and the anterior here. This is the soma, or the bodies of the motor neuron. Motor neuron. And what they'll usually do is that they send their fiber out, and they innervate, say, this is considered as part of somatic motor, and they innervate the skeletal muscle, for example. Okay, and what happened is this is the axon that will release important neurotransmitter known as the acetylcholine. They release acetylcholine. Okay. And the skeletal muscle here Well, in order to respond, they would have um, the receptor, and the receptor will sit within this motor end plate of the so this uh, the axonal terminal synapse with the I'm going to zoom up. So this is, let's say, axonal terminal, and this is the skeletal muscle. So here would be this the acetylcholine receptor. Acetylcholine receptor. Okay, so this is a cholinergic receptor, by the way. And so when acetylcholine is released, I'm just going to write ACH for acetylcholine receptor. Acetylcholine receptor will open the sodium channel. And when it's opened up the sodium channel, uh, upon acetylcholine binding, so then this membrane is depolarized, right? So it's go from negative to positive. Okay, so we call that depolymerization. Depolarization. Okay. Now this depolarization is in fact continue on, because remember, it's kind of like an action potential. So it's depolarization, then depolymerization, then depolymerization, depolymerization. Then ultimately, remember, an invaginations of the sarcolemma is the T-tubule. So the depolymerization is going to go down in the T-tubule. Next to the T-tubule is the cisternae of the sarcoplasmic reticulum. Okay, so the cisternae is part of the portal triad. Sorry, is part of the triad. Okay, so this is the T-tubule, and this is the cisternae of the. I'm just going to put SR for sarcoplasmic reticulum. So the point here is that when you want your skeletal muscle to contract, you recruit. Actually, you call upon the motor neuron. The motor neuron will release the acetylcholine. The acetylcholine will bind to the acetylcholine receptor, open up the sodium channel, and you get depolymerization all the way down to the T-tubule. And once the T-tubule is depolarized, it tells the sarcoplasmic reticulum to release calcium. Okay. So the questions in exam is that what organelles within the um, skeletal muscle fiber is hold on to the calcium? The answer is sarcoplasmic reticulum. Now, calcium is very important. It's because um, what you see here is this arrangement of actin and myosin that we saw earlier. Okay. Like this. And the hypothesis is that there is a sliding filament model. And in order to slide, the myosin, which is the bigger one, right, is grab onto the actin. Okay. If they grab on, they form a cross bridge, if you will. Okay, they act on. And then once they act on, they can cause a power stroke. So then it will slide past each other. Okay, so come forward. But normally, the myosin want to grab onto the actin, but it can't. And that is because there are proteins. I'm just going to drive here. These proteins, they're kind of like wrap around the actin. If they are wrapping around it, so the myosin cannot see the actin. There are actually two of them. They're troponin and tropomyosin. Okay. So I'm going to call this green one is troponin. Troponin. um, so these two protein, troponin and tropomyosin, is wrap around actin so that the myosin cannot see the actin. But when calcium is present, when calcium is present, calcium combines to the troponin, and in doing so, it pulls tropomyosin out of the way, right? And so if calcium, you can say, okay, so here's calcium is present, troponin binds to it, and tropomyosin now is pulling along out of the way. So now there's a cross bridges between myosins and actin. So that's why calcium is very important. And obviously, if you have too much calcium around, there's more actin combines to myosin. If there's no calcium, then the tropomyosins, um, uh, sorry, without calcium, myosins cannot bind to actin. And so I hope that you appreciate in your mind the sequence of event, right? Motor neuron release acetylcholine. Acetylcholine binds to the cholinergic receptor or acetylcholine receptor. Depolymerization of the membrane down the T-tubule, communicate with the sarcoplasmic release, sarcoplasmic reticulum to release calcium. Calcium bind to troponin, pull tropomyosin out of the way, myosins bind to actin. Is that too much information?
Yes, can you repeat that, please?
All right. All right. So a motor neuron that you see here, if you want to cause a skeletal muscle, right, a motor neuron release a neurotransmitter known as acetylcholine. Acetylcholine binds to the acetylcholine receptor. And when the acetylcholine binds to the acetylcholine receptor, it opens up the sodium channel. And you know, extracellularly, there's a lot of sodium. So sodium will enter, causes the membrane to go from negative to positive. We call that depolymerization. The depolymerization travels down the T-tubule. And because the T-tubule is in constant association with the cisternae of sarcoplasmic reticulum, and so when the T-tubule is depolarized, calcium is released from the sarcoplasmic reticulum, right? And so you just think of acetylcholine bind to receptor, calcium leaves the sarcoplasmic reticulum. And what is the functions of calcium? It binds to the troponin, which pull tropomyosin out of the way, so the myosins can bind to actin. Alrighty.
Thank you.
You're welcome. And so that's kind of illustrated for you here. um, so here we kind of know what the anatomy of what a sarcomere would look like and what different regions of the sarcomere. And it's composedly of actins and myosin. There are other, you know, there's 10 proteins that you foresee it. If I don't mention it, you don't need to worry about that. And I certainly won't ask you if I didn't mention it. Okay, now the arrangement of the of the myofilament, if you zoom in, this region's right here that you see here that have only myosins along, that will be the 8th zone, whereas this is the, you know, have both actins and myosin. So this would be considered as the anisotropic or the A zone. And over here, if this just myosin, sorry, just actin alone, that will be your I band. Okay. Now, because you zoom up, you can see that there are cross bridges of myosin. You see this? This is all myosin. And myosin will like to bind to actin, which is this, right? Uh, to 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 form cross bridges. These are the actin. These are the actin that you see here. Now, because of the surrounding the actins or these troponin, which is this component here, and tropomyosin you see here, the troponin component is the one that it binds to calcium. And in the presence of calcium, will bind to troponin, it pulls tropomyosins out of the way. Now the myosin binds to it. Okay. So make sure you know troponin is the one that binds to calcium, not tropomyosin. So at the neuromuscular junction, so here's the neural and here's the muscular. This area here is known as the motor end plate. You can see here's motor end plate. That's what the motor end plate is, this area here. Now, this area motor end plate will have a lot of acetylcholine receptor. The little blue dots here are acetylcholine. And when it's synapse, right, it's synapse, so it causes depolymerization, which is here. Acetylcholine, it's in the cleft, right? Now, if you have continuous acetylcholine release, you're going to continuous stimulation, which might be bad for you because a a type of contraction. So every contraction should have a contraction. So every contraction you could have a contraction, a relaxation, a contraction and relaxation, right? Now, when you have a contraction, because you have so much acetylcholine, you have a contraction like this, that there is no relaxation. If this is the case, it's just basically going to lock up like this. Okay, this is a tetanus, if you will. A tetanus is defined as a type of contraction when there's no relaxation. Okay, let me write that down. A tetanus is a type of contraction where there is no relaxation. Now, the question is, what causes that relaxation? That means that you have to remove the acetylcholine from the cleft. So it turns out that in the cleft, there is an enzyme called acetylcholine esterase. Acetylcholine uh, esterase. Okay, so it is an enzyme. What it does is that it takes the acetylcholine and it breaks it down to the choline and the acetyl part. So it actually it just breaks it into the acetyl part, which is CH 2CH3 acetyl part and the choline part. So in other words, you just cut it into half. And then these right here can get reuptake and it will come back to make acetylcholine again. Okay, so acetylcholine. Okay, but you have to have that enzyme, which is break it. And so if you destroy the neurotransmitter, then the muscle can be in a relaxed uh, stage. And the motor neuron, you can see here, the motor neuron open up the acetylcholine, binds to the acetylcholine receptor and open up the sodium channel. And in doing so, it's going to cause depolymerization. Okay, the depolymerization will travel down, 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 down to the T2 wheel. Remember, the T2 wheel will talk to the sarcoplasmic reticulum, opens up uh, the uh, calcium channel to release calcium. What's the question? What did a tetanus have to do with this? A tetanus is a type of contraction where there is no relaxation. Okay. The question is, what causes relaxation? The enzyme acetylcholine esterase destroy the acetylcholine. If you have no acetylcholine, then you get no stimulation. Then it'll be the relaxation phase. Does that make sense or no?
Professor, can you repeat that? Because for a moment, your mic went mute.
All right. Okay. In fact, there is an bacteria, Clostridium tetani, who produces a toxin that prevents the degradations of this uh, acetylcholine. Okay, so do you know that acetylcholine is a neurotransmitter needed for the contraction? Does everyone get that? Right. So I'm just gonna write ACH, or acetylcholine is needed for contraction. Right. So contraction. And you need to know that, right? And the acetylcholine, where does that acetylcholine come from? It comes from the um, terminal knob, the axonal terminal, if you will. So these right here release the acetylcholine, right? And then these come from a motor neuron that your mind, starting in the um, temporal lobe at these motor neuron that you find in the what they call it, precentral sulcus gyrus. Okay, and those motor neuron innervate a set of motor neuron and so forth, but ultimately it goes into your spinal cord and tell the motor neuron to release the acetylcholine, and you need that for contraction. If the acetylcholine is continuously present in the synaptic cleft, you will have a contraction where there is no relaxation. We call that tetanus. Okay, so it would be lock up like that. Now, for every contraction, there gotta be a relaxation period. And during that relaxation period is that in the synaptic cleft, there's an enzyme called acetylcholine esterase. And this enzyme digests or destroys the acetylcholine. It breaks it down to choline and the acetyl group. So it's two imagine it's like a Legos, right? Two Legos stick together, it's functional. If you breaks apart, it's not functional anymore. But that's normal physiology. The acetylcholine part and the choline part then get reuptake at the axonal terminal, takes up here, and it rebuilds it back to acetylcholine again. But if you break up the acetylcholine, then there's no more stimulation. And there's no more stimulation, you get relaxation. Okay? Does that answer your question? All right, so I hope that's answered your question. Now, um, there is um, one of the most important thing that I think you need to appreciate, and that is um, when our mind decided to cause the skeletal muscle contract, right? We can generate different types of contraction. And what I mean by that is that I can cause the muscle in my finger to contract just slightly to hold up this Tylenol. You can see that I have a little bit fever, um, to hold it up, right? But because there's enough friction to hold this um, box in place. If I don't cause the muscle to contract sufficiently, it's just going to drop, right? So it has to be just enough. That's minimum, if you will. If you want, you can say I can cause it to contract more and more and more and more and more. So what I mean by that is there's an increase in the strength of contraction. Okay. So the question is, what are the ways to increase the strength of contraction? So you know that there is contraction and there is a greater strength of contraction, the greater force, right? So there are two ways to increases the strength of contractions. I'm gonna write that down. There are two ways to increase the strength of contraction. Okay. One way, like contractions during labor, yeah, in a way, yes. There although um, laboring is a little bit different, is because it is due to the uterus, and the uterus smooth muscle. You can't tell the uterus when to contract because that is automatic. What you can do is that you can causes the abdominal muscle to contract when the uterus contracts. So you want to create it synchronistically so that to help you to push the fetus out, if you will, or this entity, or the you know this creature inside your tummy, abdomen out. Uh, the abdominal muscle you can control, but not the uterus, because that's small muscle. But you got the idea that you can causes the increases strength of the contraction. So one way to increases the strength of contraction is to, I'm going to write it down. One is to increase motor unit recruitment. Okay. Now, what is a motor unit? So this is define what a motor unit is. Now here it just shows you a simple diagram, shows you 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. This is 11 muscle fiber, okay? A motor unit is defined as a single motor neuron and a number of muscle fiber it's innervate. So if you can say that, so a motor unit is a single motor neuron and all the fiber controlled by that motor neuron. So if you say motor unit number 1, for example, how it has the external terminal splits or collateral to innervate 1, 2, 3 muscle fiber, right? Whereas motor unit 2 innervate these two hypothetically, okay? Now, if I causes the motor unit 1 to fire, you can see that these 3 muscle fiber contract. But if I recruit it, right, if I call upon motor unit number 2, so we call it recruited, so now if those 2 fires simultaneously, now you have 5 muscle fiber, right, instead of 3. So the more muscle fiber contract at the same time, the more um, the more the greater the contraction, if you will. Yeah, that makes sense, right? You get 2 muscle fiber contract, it's not going to contract as much as you have 5. So that's the motor unit. So motor unit recruitment is a way to increase the strength of the contraction. But another advantage of the motor unit is this, okay? Um, so in the future, if I tell you what, I ask you what is, um, the word is muscle tone. Now, if you go to the gym, and some people might say, oh, the guys have a nice muscle tone. muscle definition, right? That's the gym lingo. Okay? For us, a muscle tone is defined as uh, a state of partial contraction. Okay, so that's what muscle tone is. Now, what I mean by as partial is this. Now, if motor unit contract, motor unit one contract, only three muscle contract, right? So that's partial, not full. Full will be 11. Okay? Now, for you guys who's sitting like I am sitting, I want my neck to hold up, right? Or perhaps the muscles in my back to contract, um, so that at least I can sit upright, or to hold my head up, right? One of the important concept too, another one that I think is important to note is it's known as muscle fatigue. Now, muscle fatigue is like you can say it's uncontrollable contraction. So you can contract, particularly skeletal muscle contraction. It get to the point fatigue, en français fatigue. I think some of you might speak France. Comment on dit tire en français? Does anybody know? How does one say tire in French? What? You don't have any
Fatigué?
fatigué or fatigue, oui? Right?
Oui.
So fatigue means tired. And so when we say muscle fatigue, is that the muscle is tired. So you tell it to contract, it's not going to contract anymore. So that's what we mean by muscle fatigue. And the last thing you want to do, right, is is obviously if you're running, you can have muscle fatigue, but you sit in the class, you don't want the muscle in your neck to go into fatigue, and then you don't want to contract anymore, right? And so the way that um, we have muscle tones in our neck, and that is there's a partial contractions of the muscle, enough to hold up your neck, your head, right? But this idea is what we call it the alternative usage of motor unit. Okay, let me write that down. Alternative usage of motor unit to ensure a continuous state of partial contraction. So I hope you guys appreciate this idea is that the alternative usage, meaning that if you causes this motor unit one to contract continuously for 3 hours, these three muscle, right, for 3 hours will undergo fatigue and it's not going to respond anymore. And so what you do is you say, okay, why don't I use the motor unit number one now? Three muscle, that's partial contraction, enough to hold up my head. If that is tired, let us relax. Let me call upon motor unit number two, right? Or maybe unit number three, and so forth. So you alternate usage of this motor unit to ensure that you always have this state of partial contraction continuously so that you don't undergo muscle fatigue. Is that the anyone clear with that? Okay, good. No. Um, what is the question?
Can you
Uh,
please repeat the last sentence you made? I just wanna. about each arm, the one tone is tired, you let the two carry out the stuff.
right? So the word muscle tone means partial contraction. By partial contraction, meaning that not all the muscle contraction, but only some of it, right? So like three muscle. So in fact, the muscles in your neck right now is in muscle contraction. It's only partial contraction. You can recruit more motor unit to have a stronger contraction, like that, right? That's a stronger contraction, right? But you don't need that because if you continue doing this, you're gonna get muscle fatigue, right? But the reason why you can hold your head up for 3 hours, because not the same muscle that contract that 3 hours. Is only one group work now, another group, and another work group, and another group. So it's almost like you always asking three students to stand guard, right? You can't ask three students to stand guards for 3 hours, right? So we say, okay, how about three students guard for 15 minutes? You tire, you rotate out. Another three students. And then if you tire, another three students, right? 3 out of 25 students is partial contraction. You see that? If if you want more contraction, you will cause three to stand guard, and another three, and another three, another three, until every muscle contracts. That's the maximum contraction. If the maximum contraction to a point where you cannot contract anymore, that is known as muscle fatigue. I already told you already one way to increase the strength of contraction. I haven't told you the other one yet, and I'm gonna tell you in a little bit. So, um, so then to the summarize is what I'm saying. Uh, Before we go on a break, is this idea? Let's call it the excitement contraction coupling. Okay. Now, the key here is that the muscle impulse causes the sarcoplasmic reticulum to release calcium, right? So we know that upstream of this will be the depolarizations of T tubule. Calcium binds to troponin, changes the shape, causes the tropomyosin pull out of the way. Actin is exposed, and then actins and myosin combine too. So you can see here is that the head or the catalytic site of the myosin is actually an ATPase. This is very critical. This is the area that has that does the binding, but use ATP, and we need to know where did why is ATP is important. But suffices to say that in the presence of calcium, okay, calcium binds to troponin that you can see here, pull topomyosins out of the way. So this is supposed to be this little, now you see the little dot, the red dot here, right? So then now myosins can bind to that attachment. And what you're going to see, there's going to be a power stroke. So this is known as a power stroke. And then I will tell you there's three area why ATP is important. One is that ATP is needed to um, is important for hydrolysis of this thing called power stroke. Um, or sorry, actually, it should go this way. So when the presence of calcium binding, the um, myosin is going to attach this to actin, and then the hydrolysis causes the power stroke. ATP is needed for the detachment, okay? Because without ATP, the myosins will stick. And we're going to talk about a little bit in the context of rigor mortis. And then the hydrolysis of ATP recoccus cell, and then you get the next cycle. Okay. All right. So why don't we take a 5-minute break? I'm going to stop here,